The skeletal system has a number of roles. Obviously here you can see that the bones are providing support for the softer tissues, and then protection as well. So the delicate brain is protected by the cranium, the heart and lungs by the rib cage, the delicate pelvic organs by the pelvis, the spinal cord by the vertebral column, etc. The skeleton is involved in movement. Now, I know you might say, no, that's something that muscle does. But say the muscles of the arm can only cause forearm flexion because, say, the biceps attaches to the bones of the forearm. And so thus the bones of the forearm move when the muscle contracts. And so muscles are involved in, the bones are involved in movement as well. There are other roles of the skeleton as well. They help us balance out calcium in the body, as I'll discuss later. There is marrow, which can occur in the spaces of bone. And this, so here you can see some spaces, so marrow can be found here. And in this marrow, we can make blood cells as we have stem cells which can then respond to hormones and make various kinds of cell types. So here you can see red bone marrow under the microscope where red blood cells, white blood cells, and platelets are being made, millions per second. So that's obviously an important role. And then in some regions, there is adipose. And so here you can see adipose cells in what's known as yellow marrow. When we were infants, all of our bones had red bone marrow, but then that became restricted. So for example, in my arm and in my leg, the only place where I have red bone marrow anymore is at the proximal epiphysis of the humerus or the proximal epiphysis of the femur. But if I were to get cut and bleed a great deal, some of that yellow marrow could temporarily become red marrow. Now, this first chapter is on bone tissue and cartilage tissue, and then the upcoming chapters then go through the anatomy of the bones of the arms, the legs, the skull, etc. And so there are two types of tissue in the skeletal system. There is cartilage, and as we'll see, there's different kinds of cartilage. And so some of these, cells look a little bit different from others. So there are different kinds of cartilage. And then there is bone tissue, okay? And so one of the things I struggle with my students is we often picture bone or cartilage even like this. Because if you study in a lab, you might actually have real bone and cartilage sitting on the table at some point. But this isn't real bone or cartilage. So this is dead, remnants of what bone was. So, the bones that sit on a table, they might actually be rather brittle. I mean, this is actually rather brittle. But the living cells and tissues are very, very different. Living bone has the same strength as steel reinforced concrete. But if you see a bone lying on the table, you're thinking how brittle it is. If I smashed it like this, it would break. And that's because that's not real bone. In life, these are real living tissues with real cells. So here's cartilage. You can see that there are real cells here. Cartilage there. The cartilage cells can occur in spaces known as lacunae, and actually the components of this space are actually excreted by the cell. So here you see the cartilage cell actually synthesizing the components which will surround it here, so that it is isolated from the surrounding matrix. Cartilage is avascular. Notice there are no blood vessels here, whereas bone is very vascular. These openings here, and here, these are for blood vessels. So They are similar in that they are both alive, but then they are different. Cartilage is avascular, bone is vascular. They are both solid connective tissues. So around this osteocyte, notice that there's a solid matrix surrounding the cell. Here in the cartilage, notice there's a solid substance surrounding the cells. So there are both similarities and differences between cartilage and bone. Now, these tissues can grow in one of two ways. Cartilage will grow in both ways, but bone will only have one of these features. So notice here that here we're looking at the outside of the piece of cartilage, which I'll call the perichondrium, and I'll better explain that in a minute. And notice that the chondroblasts, these are immature cells which can make cartilage. They are laying down additional cartilage from the outside. So because the chondroblasts are laying down more cartilage, the cartilage is becoming wider. There's more cartilage here than there used to be. That is what is known as appositional growth. Kind of in the same way that tree trunks get wider, you continue adding more wood to the outside of the trunk. That's appositional growth. But cartilage can also then grow from within. So here you see chondroblasts within these spaces known as lacunae. They can divide, and they can then make a cartilage matrix between them. And so then as cartilage then accumulates between these cells, the cartilage is now getting wider from within. This is known as interstitial growth. So cartilage can grow two different ways, both through interstitial growth and through appositional growth on the outside, whereas, as we'll see, bone can only grow in one of those two ways, appositionally. Okay, so here you can see, cartilage can grow appositionally. Cells on the outside can end up now in the inside. They make the materials in the lacuna which surround them, and then they can continue to divide and have the cartilage grow interstitially. The three types of cartilage which we find are hyaline cartilage, the main class, and then two others, fiber cartilage and elastic cartilage. Hyaline cartilage is the way that most of our bones form. So the bones of my arm or leg, they started off as hyaline cartilage that gradually became bone, but I still have cartilage at the tips. So at the very ends of these bones, where, say, the bone of the humerus rubs against the bone of the scapula, it's actually cartilage from the humerus articulating with cartilage of the scapula, so that we don't have bone-on-bone contact. Here you can see hyaline cartilage where the ribs meet the sternum. These are called costal cartilages. The trachea and larynx are made of hyaline cartilage, and that's what we'll see under the microscope here. This is hyaline cartilage. So notice that it is a solid connective tissue. There is a solid matrix here. There are living cells. Cartilage is a living tissue. There is a solid matrix, a gel known as chondroitin sulfate. And there are also collagen fibers here. Collagen composes, up to 40% of cartilage. It's the most abundant protein in the body. And so thus, with specialized cells, extracellular protein fibers, and a ground substance, cartilage is clearly a connective tissue. In addition to the hyaline cartilage that you just saw, there is also another kind known as fiber cartilage. We find fiber cartilage between the two pubic bones at a place known as the pubic symphysis, or the pubic symphysis. In the intervertebral discs between vertebrae in the vertebral column, we find fiber cartilage there. And when we break a bone, during fracture repair, we make calluses of cartilage, and it is fiber cartilage, which we find here. There are other sites as well. So for example, between at the knee, we have structures known as menisci, which are made of fiber cartilage. So here you can see fracture repair there. Finally, there is a much more flexible kind of cartilage known as elastic cartilage because of elastic fibers that one can find there in the external ear. We have a very flexible cartilage there, but also in the epiglottis, this piece of cartilage which can close over the glottis, which is the opening to our airway when we swallow. So if we don't want a bolus of food to then pass into our trachea where it could cause us to suffocate, the epiglottis closes over the glottis, and the epiglottis is made of elastic cartilage and a very important part of it. So while we often think primarily of skeletal tissue when we think of the skeletal system, obviously cartilage plays an important role as well. Now, both bone and cartilage have an outer covering. If it is bone, we call the outer covering the periosteum. And if it is cartilage, we call it perichondrium. I'm sure you've gotten this point by now that chond is the root for cartilage. So chondroblast, chondrocyte, perichondrium, these obviously refer to cartilage, while osteoblast, osteocyte, periosteum, these refer to bone. So there is an outer covering in bone tissue known as the periosteum, and cartilage would have something similar. And this would include here in green the osteoprogenitor cells. Osteoprogenitor cells, they can divide and become either chondroblasts to make cartilage, if that's what you need, or osteoblasts and make bone. So cartilage and bone are connective tissues which are made by the descendants of the same stem cells. Osteoprogenitor cells in the outer coverings can result in the production of either bone or cartilage. Bone can actually then have more of this in what's called endosteum, deeper inside the bone around, say, blood vessels, but I'll mention that again presently. Okay. Cartilage is a connective tissue. Likewise, bone is a connective tissue. It has specialized cells. Like I've said, these include stem cells, which can divide. These stem cells are called osteoprogenitor cells. These can become either chondroblasts or osteoblasts, depending on what the body's needs are. And as we'll see later in fracture repair, sometimes we fill in with cartilage, sometimes with bone, depending on whether there's a blood supply. Osteoblasts are the immature cells which make more bone, and this occurs through appositional growth. Say the osteoblasts in the periosteum then lay down new bone. Now, osteocytes are the mature cells which exist inside the bone matrix, and they do not make bone because there would be nowhere for the bone to go. Notice that this is a solid connective tissue. If you were to try to make more bone, you simply couldn't force this bone tissue farther apart. It's too strong. So osteocytes would simply maintain bone, and they'll change their shape by growing cytoplasmic extensions which pass through channels known as canaliculi, as we'll see. There's also another kind of cell in bone derived from blood cells. Now, usually monocytes, they say, eat microbes, but a bunch of them can smush together and form a big multinucleate cell called an osteoclast. And instead of using lysozymes and acids, et cetera, to eat microbes, it dissolves bone. So osteoclasts are another class of cell in bone tissue, which dissolve bone. So there are specialized cells. There is collagen, which isn't surprising. I mean, this is a connective tissue, and connective tissues have extracellular protein fibers. Like collagen, the primary extracellular protein fiber here. And about a quarter of bone is actually collagen. Now, you don't see it normally because you're, perhaps taken by the hard ground substance in the matrix. But cartilage has collagen in it, can be up to 40% collagen, and here bone. If you were to take all of the calcium salts away, you would be left with this. This is decalcified bone. And you can see that, in addition to the cells, there is stuff here. This stuff is the protein collagen. So bone is a connective tissue, and collagen, the most abundant protein in the body, makes up the extracellular protein fibers in bone. If your skeleton was just made out of collagen, it would be too flexible. If it was just made out of calcium salts, it would be too brittle. The two of them together form this incredibly strong ground substance known as hydroxyapatite, which has the same strength as steel reinforced concrete. All right, and so bone, in addition to cells, has both collagen fibers as the extracellular protein fibers, and then the ground substance of hydroxyapatite. Okay, now, as we look at bone, you'll see both osteoblasts, and these can be present, in that periosteum and endosteum, where it can lay down new bone. Once osteoblasts are left behind in the bone matrix, they then become osteocytes. Osteocytes no longer make new bone tissue because, as I said, there would be nowhere for this bone tissue to go in such a solid matrix. Now, when we speak of bone, one of the first things that we can do is say, well, let's classify bone into categories. And then say lab, you might say there's a difference structurally between, say, a long bone and a regular bone, et cetera. When we look under the microscope, we could distinguish between two types of bone tissue, what's called compact bone and spongy bone. So this is compact bone, all right, where most of the spaces are filled in with bone tissue. And compact bone is composed of these structures known as osteons. What's an osteon? Well, this is an osteon. There's a big hole in the center known as a central canal. And in this central canal, blood vessels, a small artery and a small vein would pass. Around these blood vessels in the central canal, there would be cylinders of bone salts. These would be concentric cylinders. So there would be one cylinder right around these blood vessels, and then another one around that, and another one around that, and another one around that, etc. So concentric lamellae. Now, in these lamellae would be these cells known as osteocytes. So notice these dark spots here. These are the osteocytes in mature bone. And as we'll see, if you look closely, there are these thin channels which unite them. Because obviously these cells are very close to the blood supply, and they can very easily, get their nutrients and gases. But the cells which are farther away, how would they get their oxygen or glucose or remove their waste? Well, there are these small channels known as canaliculi, which unite the osteocytes throughout the bone with the blood vessels here in the central canal. So here you see compact bone. Once again, there's a central canal for blood vessels. And then we have concentric circles of bone salts known as lamellae, and canaliculi can unite the osteocytes between them. In this next type of bone tissue, notice that the bone is not as solid. All right, there's lots of spaces between it, and therefore this is what we call spongy bone. Now, compact bone doesn't have, those spaces, and the osteons all run in the same direction. And so in this, say, the shaft of a long bone, the diaphysis, the osteons are all running vertically, which is now good because when we jump up and down, all of the osteons are oriented in the direction where the stresses are coming from. But at the ends of the long bones, the epiphyses, notice that we don't have osteons. Instead, we have these short trabeculae, these short struts of bone. And this means that stresses can be withstood from many different directions. So, when you're moving or dancing or whatever, as the center of gravity shifts, then how stresses are reaching the epiphysis of the femur would vary. So we need a different type of bone there, which can withstand stresses from multiple directions. So compact bone is found on the surface of all bones, as you can see it found on the surface here, but it also makes up the shaft or the diaphysis of the long bones. In contrast, at the epiphyses, the ends of the long bones, we find this spongy bone with its struts going in many directions. And here also then we have spaces, so you can have, say, a red bone marrow here. So under the microscope, you can see different types of bone tissue. There is both compact bone and spongy bone. We can also talk about how bone forms. Now, before we talk about how bone forms in two separate categories, let me just talk about something that they share. Bone can only grow appositionally. So here you see osteoblasts of the periosteum. These osteoblasts can lay down a new front of bone. Bone cannot grow interstitially. And as bone grows appositionally, some osteoblasts can get left behind and they can mature into osteocytes. This bone front can grow around collagen fibers. So on the surface of a bone, there is collagen. This could be connected to the ligament from an adjacent bone, or the tendon from a muscle which attaches to that bone. As the osteoblasts lay down new bone, the new bone tissue now incorporates this collagen with it. So that's why ligaments and tendons can have such strong junctions with the bone. So if I were to fall, I could break my collarbones. But the tendons, which hold the arm muscles to the bone, or the ligaments which attach the bones to the joints, they might still be intact. Because as the bone grows, it can incorporate collagen fibers from neighboring ligaments and tendons so that it has an extra strong attachment point. So when my biceps brachii pulls on my radius, the radius comes because the collagen from the tendon of the muscle then goes actually into the bone and become parts of it. And then finally, blood vessels, which exist on the outside of the bone, can then be incorporated onto the inside of the bone as the bone grows around them in appositional growth. So as bone grows appositionally, a number of structures can get incorporated inside. Osteoblasts can become osteocytes. Collagen fibers can get incorporated inside for an extra strong junction. And then in addition, blood vessels can get incorporated inside. As an adult, the bone tissue in my body is the same. So, this bone tissue is the same as this bone tissue. But they formed in two completely different ways. And so this next then distinction is the description of the two different ways in which bone can form in the body. Some bone forms through what's known as intermembranous ossification. Membrane just refers to some region of connective tissue, such as the dermis. Intermembranous ossification just refers to the fact that we can make bone anywhere. So as a fetus, I have these stem cells, these mesenchymal stem cells. Stem cells haven't quite decided who they would like to be when they grow up yet. they can then differentiate not into, say, fat cells or not into fibroblasts. They could differentiate into osteoblasts, okay, or osteoprogenitor cells, which then become osteoblasts. And so now we, in the middle of what we had as a dermis, now we have the cells which make bone. They're going to be wrapped in a periosteum. And then the osteoblasts here, they can then just make more bone and make more bone, and then the bone expands. So intermembranous ossification is just formed in the middle of a membrane, like the dermis, and therefore can result in dermal bone, where just, there was, a regular connective tissue, and now it has bone in it. And so when we look at a turtle shell, that's dermal bone. There's dermal bone in the hide of a crocodile or in an armadillo. All right, so you can put, bone in any, region of the dermis. We put it in our skulls. So our skull and part of our clavicle, these are the only regions where we have this dermal bone, which formed in a say in the dermis through intermembranous ossification. Obviously, it can grow, and as it grows, osteoblasts can get converted into osteocytes. The ligaments from neighboring tendons, the collagen from neighboring tendons and ligaments can get incorporated as well, and the bone can grow around blood vessels. Now, if you're asking why is our skull and our clavicles special, that they are made in this way, well, it kind of dates back early to vertebrate history. The first vertebrates which had bone were jawless fish, and they made it in their heads. So in the first vertebrates with bone only existed in the heads. The rest of the skeleton was cartilage, which would later become bone. When you were a fetus, you put bone in your head, and then the rest of your skeleton was cartilage, which would rather become bone. So that's why, the bone in your skull forms one way. Not every part of the bone in your skull forms through intermembranous ossification, just most of it. And the reason your clavicle's involved is because in fish, the pectoral girdle is attached to the skull. There is no neck. And so this bone then had a similar form of ossification to the other bones of the skull. Most of your skeleton, say the bones of your arms and legs or your ribs or your vertebrae or your hips, they then form a different way. They form from a cartilage model first. And so the mesenchyme differentiates, not making osteoblasts, but rather chondroblasts, which lay down a cartilage model, which might look an awful lot like what the bone will ultimately be. These chondroblasts make cartilage. They develop a perichondrium, all right, an outer wrapping, which has, chondroblasts in it. Okay, and so at a certain stage of fetal development, if you were to look at an ultrasound, you would say, oh, there's the skull bones, and you would say, oh, the frontal bone, that is made of bone. The parietal bone is made of bone. But you wouldn't say, oh, that's the arm bone or the leg bone, because these now are cartilage. They have not yet become bone. So much of our skeleton undergoes this endochondral ossification, where at first it starts off as cartilage tissue, which will later become bone. Now, at a certain point then, blood vessels approach. The periosteum will lay down osteoblasts, and the chondrocytes begin to deteriorate. They hypertrophy, they enlarge, and the cartilage calcifies. That's a negative development for the cells in the cartilage, because remember that cartilage is avascular. Because it's avascular, these cells are relying on the gel-like matrix of chondroitin sulfate in the cartilage for diffusion. They need gases and glucose to diffuse. Now that's hard enough as it is. That's why cartilage doesn't heal very well, because in a normal cartilage, it's hard for this diffusion to occur. But if this area starts to calcify, then this becomes much harder still because now the matrix is too rigid and no oxygen is passing. So the cartilage will die and deteriorate and the bone fronts will advance. So because osteoprogenitor cells can become either osteoblast or chondroblast, here in what was the perichondrium, the osteoprogenitor cells produced some osteoblasts, and now we have a periosteum with osteoblasts. And osteoblasts, as they make bone, now the bone can expand through what's called appositional growth. And so just growth from the outside, the bone gets wider, and this bone front becomes longer. Once again, this is growth from the primary center of ossification. As it does so, obviously, osteoblasts are getting incorporated inside. The bone front is growing around collagen fibers and blood vessels as well. Now, you might think that once we start making bone, that this bone could then just proceed until the ends, and now the humerus is now completely bone instead of its original cartilage. But that would be a problem for two reasons. One, if during fetal development or during infancy we turned the humerus into bone, one, it would be done growing, because bone cannot grow from within. And once the humerus became solid bone, whether it was this big or this big, that would be the end of the humerus. It would never get to be this big. So we don't want this bone front to reach the ends just yet. But plus the fact that would mean that at the ends of the bone, bone would then articulate with bone. Bone would rub on bone, and we wouldn't like that. It would chip. It would, you'd have these little sharp fragments of bone. It would squeak. We don't want bone rubbing on bone. We want this cartilage at the very ends of the bone to stay, so that at a movable joint, it's cartilage rubbing on cartilage. So from this primary center of ossification, we do have bone advance, but we don't want it to reach the ends yet and completely convert the structure to bone. So we develop what are called secondary centers of ossification, where once again, the cartilage will hypertrophy, nutrient arteries will penetrate, the osteoblasts come in, the area will begin to calcify, and then we have now new bone fronts, not the primary center of ossification that started here in the shaft, but now secondary centers of ossification in the epiphyses, and now then they can grow. And it's nice to have bone tissue at, in the epiphyses because, as a child, you were jumping around, you were playing sports. And so now we have this strong bone tissue to, help, support that, etc. But note that between the primary center of ossification and the secondary centers of ossification, we have a band of cartilage. This is known as the epiphyseal plate, sometimes called the growth plate, and it is essential. If a child has a fracture and it affects the growth plate, then that's, concerning, because this could affect, potentially the growth of the entire limb and its resultant length. So while bone can advance, cause the cartilage to calcify, the cartilage dies and the bone advances, cartilage can grow interstitially. It can grow from within, and it can push away from the bone. And this next image, that's what you're looking at a slide of the epiphyseal plate. You will see here is the cartilage between two regions of bone. There is the bone in the epiphysis, which is resulting from that secondary center of ossification. And then there's the bone in the diaphysis, which arose from that primary center of ossification. But here between the two, we have cartilage. Now, this cartilage here isn't very active. The bone front at the epiphysis isn't really advancing. So that's referred to as resting cartilage. But here in the center, we have cartilage cells. You may notice that they occur in these rows because cells divide. And once they divide, because cartilage can grow interstitially, they then make cartilage between them. And then they divide again and make cartilage between them. And then they divide again and make cartilage between them. And so notice the cartilage between these two cells which arose from cell division. Notice the cartilage between these two cells, which arose from cell division. So we have zones of proliferating cartilage where the chondroblasts are undergoing mitosis, and then as they grow interstitially, cartilage develops between them. Now, as we near the bone, the cartilage starts to deteriorate, the cells swell, all right, they'll start to break down, and then there is a zone where the cartilage is calcifying thanks to the advancing bone front, which is using, additional calcium to produce, calcium salts. Once again, this is bad for the cartilage because this avascular cartilage relies on diffusion to get nutrients to the cells. And once we calcify the cartilage, then it is harder for nutrients to reach, the cells of the cartilage. So this is a growth plate or epiphyseal plate. It is a band of cartilage between two bone fronts, the bone in the epiphysis, which came from the secondary centers of ossification, and the advancing bone in the diaphysis or shaft from the primary center of ossification. Now you see how much proliferation is going on here. These cells are just dividing and dividing and dividing and making more cartilage between them. And then this allows the cartilage to grow. So the cartilage grows and the bone catches up. The cartilage grows and the bone catches up. The cartilage grows and the bone catches up. And you ask, well, what are we accomplishing? Well, we're growing. There's now more bone than there used to be. So as long as this band of cartilage exists, it can push away and the bone can actually become longer in length and you can become taller. When, however, puberty hits, the hormones of puberty will then tell this bone front to start growing faster. When it does, then the bone, influenced by estrogen and testosterone, these are hormones which affect bone growth, can then have the bone front advance and advance and advance until the last of the cartilage calcifies, it dies. And now once you have the bone front in the diaphysis fuse with the bone front in the epiphysis, now the bone has reached its final length. It will not get any longer. You may see an epiphyseal line briefly, as I'll get to in a second. Now, before we get to the epiphyseal line, notice that there is still cartilage here and here. So the bone did not completely overtake the cartilage and reach the end. This structure was originally entirely cartilage, and now we still retain a little bit of cartilage here at the ends of the bone. And now as a result, at a joint, we don't have bone rubbing on bone. You have cartilage moving on cartilage. And when we talk about joints and articulations, that is an advantage. Now, there are times where bones can fuse in our skeleton. So your sacrum, your os coxa, even your scapula with the separate coracoid bone can fuse. But now also here, where the epiphyseal plate once was. Here you can then for a while see an epiphyseal line, that when the cartilage dies, an epiphyseal line will form where these two separate bone fronts. So during childhood, say this is a separate piece of bone than this piece, and they are separated by a band of cartilage. Some bones are complex, like the femur. So you can get, for example, at the proximal end of the femur, you can get multiple secondary centers of ossification, say one here, one here, one here. And therefore, once the bone catches up, we see what are called epiphyseal lines where these meet. Now, different parts of your skeleton finish their growth at different points. So for example, toes may, finish growing early. Your long bones may finish growing in your late teen years, but other bones later, including, maybe the xiphoid process doesn't ossify and fuse to your sternum until age 40 or so. And so when these cartilage models become then bone to the point, that they will. This can vary throughout the skeleton. Once again, however, when we talk about bone, there are two different kinds of bone when we look under the microscope: compact and spongy. And then there are two different kinds of bone when we talk about how bone forms. So there is both intermembranous bone, which forms just, as a center of ossification forms in the middle of a membrane like the dermis. And then there's endochondral ossification, where we start with a cartilage model of the bone, which is gradually then converted to bone.